Yes. So, good morning, everybody. Probably a few more people will join later, but we'll just start. I'm Sara Graziadio. I'm a senior methodologist in a Newcastle MIC, and I will introduce the MIC in the next few slides. And Rachel, do you want to introduce yourself?
Yeah, hello, I'm Rachel Dickinson. I'm the Research and Development Study Manager for the Newcastle MIC. I also kind of lead on communications, and I've been kind of organising this series of webinars along with Sara.
Yeah, so thank you, Rachel, for that. It was a lot of work. And this is the first time we're doing it online. It used to be a face-to-face session, but we decided not to give up and still give the opportunity to actually share this information with you, especially in this time. It may be helpful actually to know what is the evidence development pathway for new diagnostics. For COVID, it's probably going to be a bit different, but we have to remember what is the standard practice and what is the robust way to do generation of evidence in diagnostics anyway, especially now. So the format it will be presentation. I will have 3 polls where I'm asking you questions, so we can involve you directly in the conversation. You can post questions that you've got for me on the chat, and then or you can at the end raise your hand. We will collect the questions, but it's going to be just Rachel will see them as a host, and then we'll answer to the questions at the end. Possibly Rachel will remove the ones the duplication and things like that. So Rachel, do you want to explain about the feedback?
Oh yeah. So at the end, in the next couple of days, as Sara said, we'll share links to the slides and also to the recording, and at the same time we'll send a link to a short feedback survey, because we're always looking at new ways to improve our delivery of these sorts of sessions and the sorts of topics that people might want included. So if you can fill that in, that will be great.
Thanks, Rachel. So there is a hashtag, #HaveDev4Diagnostics, for the whole series. So if you want to follow and join the conversation there, you're very welcome. And I think we can start now. Are we, everybody? Hopefully. We gave the 6 academic minutes, so. So today is just an introduction, really, quite a high-level introduction about the whole series, and so the aims is really to understand the role of diagnostic testing and the evidence that we require to facilitate adoption of a new test in the NIHR. Really, the hope is that we can develop and make sure we've got a common language. Sometimes in diagnostics, that's still not the case, and it's really sometimes a problem in terms of making sure we're all on the same page and you know where we are coming from, you as developer, and we can have the same conversation using the same terminology. So I'll try to set up the language today a bit, and obviously every next webinar will fit within the evidence development path we will describe today. So hopefully at the end of this series that's going to last for a month, it will be clear what we mean by evidence development pathway. Finally, we want to explain who we are, what we do at the Newcastle MIC, and who we work with, because we definitely don't work in isolation. We've got a lot of collaborators, and there are also some of the people giving the talks, and that's been the case since last year. And this is really an opportunity to show how we work collaboratively with the university and the trust and the region. So we start soon with the poll to understand a bit more who you are and why you're interested in this. Rachel, if you can put it up, the question, you should be able to see it on your screen and answering it. So we can understand where you're coming from. We know that we've got quite a good representation of both universities and industry. So yeah, that's very interesting audience. That's the people we work with usually. So I can see people are voting. It's good to kind of have a starting point, knowing our audience a bit more. A lot of people interested in the subject, not developing and evaluating tests yet, but so we tend to work with developers and we collaborate with the evaluators. So it's interesting to see who is joining us today. Okay, I think we can end the poll. Oh, we've got 5 people more. So yes, that's very useful to know. Hopefully this will be helpful for you today who are developing and evaluating a test in the process of it. So thank you for this. Rachel, can you remove it? Okay. So now I'm going to tell you who we are. So perhaps you've heard of us already, as if you've signed up today, but we are one of the 11 NIHR MICs. We've been funded in 2013 as a diagnostic evidence cooperative with the aim of generating evidence for in vitro diagnostics. In 2018, we've been rebranded as a MIC, and we've been the DEX and what they were called previously HTCs that were joined together to simplify the landscape of development and generation of evidence for medtech and in vitro diagnostics. So MIC now means medtech and in vitro diagnostics cooperatives, and there are 11 of us, but the previous decks focus on mainly on evaluating diagnostics, in vitro diagnostics, and we are in Newcastle, Leeds, London, and Oxford, while the previous HTCs mainly focus on development of medical devices. So we've got slightly different aims, but yeah, we try to work collaboratively, especially with the other decks. For example, at the moment for COVID research, we are about to launch an initiative that sees the X-DEX together to help the government to do robust evaluation of new diagnostic tests. So this is our team. We are quite a few people now. John Simpson is our director, and Andrew Sims is our deputy director. Joy and I are the senior methodologist. You will meet Kasha. She's got a presentation on public and patient-to-public involvement and engagement later on. Rachel is our R&D study manager and our PM, and these wonderful methodologists are really the team who delivers the work every day. You will see some of them delivering some of the workshop in this series. So we've got 3 main clinical teams. Apologize for these horrible pictures that we need to fix. The main team that we've really been working on in the last year has been infection diseases with Ashley Price. We've done a few studies on sepsis, flu, and now recently we've been quite involved with COVID-19 testing strategies. We've got an aging and long-term condition with Miles. Especially we had the focus on stroke, COPD, and then more recently Alzheimer's and Parkinson's. And then we've got personalized medicine theme with Nick Reynolds. As I said, we don't work in isolation, and we are just in the middle of a very big network that can help and support companies and clinicians and researchers from the stage of discovery invention up to dissemination. We work in close collaboration with the HSN here in the region, and also we've got a few projects with the HSN Oxford, and here in Newcastle there is also the Innovation Observatory that helps with the horizon scanning. So, you know, there is support that we can provide. I'll start with, as I said before, saying some of or deciding about the language and some of the definitions that can be useful when in the conversation and to understand the context of what we are doing and the evidence development pathway. I'm sure all of you know what's a diagnostic test. So you've got patients with symptoms, the doctor doesn't know what's wrong with the patients, and then or the diagnostic test or one diagnostic test. If the results are positive, then you've got a treatment, but if they're negative, then it may be that the patient does nothing particularly wrong with the patient, but if symptoms persist, then you need to pass the test. But we know that there isn't just diagnostic tests, and the clinical tests that we can use are multiple. So I've got another question for you, and then we will go into seeing what kind of clinical tests that there are that we can evaluate. Rachel, if you want to launch the second poll. And so here it's a bit longer to read, but which one of these sentences is correct? And is to understand screening tests, diagnostic tests, monitoring tests. And then I'll tell you some of the definitions that we use. % of you voted. Okay. We've got a very informed audience. So yes, the second and the third one were the correct one. So very good. We'll introduce now some of these definitions, and then we'll move to the evidence development pathway description. So as I mentioned, we've got 4 main types of tests: screening, diagnostic, prognostics, and monitoring. And the main difference is what is the patient population that the tests are used on, and what kind of decisions and clinical decision they will inform. So for screening, you are testing asymptomatic patients, and the main aim is to inform further screening, further testing. Diagnostic is for symptomatic patients and inform mainly treatment, but obviously that's very simplistic, and there are a lot of other clinical decisions that are informed by diagnostics. For example, could be isolation or PP use. Prognostic, yeah, we've got a news to chart, and so they don't have to be just in vitro diagnostics. Sometimes you've got charts, could be also tests, and they're used on symptomatic patients to inform risk of deterioration usually, or and they're very helpful to organize the hospital flows. Then monitoring tests that are on patients who have been diagnosed and who've been treated, but you want to understand what is the efficacy of the treatment on that population. So let's focus now on the evidence development pathway. That's the main topic of today of the whole webinar series. As you hopefully will see, every talk really fits and explain one aspect of this evidence development pathway. I just wanted to point your attention that we've got a paper that's been just published in March and is actually covering, I think, almost all the points and also a bit more than what we're going to cover during this webinar. So if you want to know more, have a look to the papers, and you can have you can see we're also covering the PPI aspect of it. For example, so this is the main picture that we've got in the paper, and that we'll be using during this series for describing the evidence development pathway. Can see it quite complex, but I will go through each of these aspects in the next slides. So now I just want to really focus on a few main elements. Obviously, this describes and summarizes the journey between from discovery to adoption of a new diagnostic test. Unfortunately, it's never so linear, it's never so simple, but it's good to try to simplify the process so that you can find where you are in this journey and where your technology is in this journey. It's not that linear. Sometimes you've got loops, you have to repeat step, things do not really, usually, especially at these early stages, you've got a prototype, it doesn't work, you have to build it again, and so on. But the main steps are these. Sometimes, for example, you can not have a diagnostic accuracy phase. For example, if you've got a really terrible reference standard and move, and if you've got a very strong test on the analytical validity phase, you've got some good evidence of safety, and you could move in clinical utility phase directly. It's quite rare, but can happen. And sometimes on the other, instead, you could avoid the clinical utility phase if you've got a very big, strong evidence built through the diagnostic accuracy, and you can move into an adoption phase where you've got designs that are more real world. So it's not, they're not all the steps always all necessary, but you need to be very aware of what they are so that you can decide if one is not necessary or if one is actually more important for your technology. The other thing I wanted to point is these lines here. What do they represent? They represent the uncertainty level. So if you're starting in this phase, we really don't know if we've got a test. I mean, how does it work? Does it really work? The level of uncertainty is really, really high. The more we go forward with the journey of generating evidence, the less uncertainty we've got. And this is a very important concept. It seems quite intuitive, but it's really at the basis of statistics, and this will be discussed in the next talk about diagnostic accuracy studies, but it's also an important concept in health economics. So I'm sure Joy and Woven will cover these again in the next webinar on health economics. Another point I wanted to bring your attention to is these studies here at the bottom. I'm not going to kind of spend a huge amount of time on these, but it's to show you that the evidence that you require to get to adoption is not just focused on the clinical studies. You need more than that. Some evidence is more qualitative, so it's linked to barriers and facilitators adoption, usability, and human factors. So you don't just want to understand if your test works, but you want to understand if it's usable, if it can be, if it's feasible to use in the setting and with the end users of interest. Do you need to have a patient to use it? You need different a different test than if actually you're waiting for, you're expecting a clinician to use it, perhaps in a very controlled environment like the ICU. So you need to keep that in consideration, and you need to have studies that assess the usability during the whole duration of these initial phases when you still have got a prototype stage. Barriers and facilitators adoption is also an important aspect. Sometimes some barriers can be identified in early stages, and that means it's more likely that you can solve some of these issues, and that means that then you've got better chance to get to adoption at a later stage. Facilitators, sometimes, for example, you can have the possibility to have champions that could promote your test with clinicians. There could be factors that you want to, that seems to be a strong selling point for your test that perhaps you didn't think about. This can come, for example, from clinicians that see your test in a different light from what you see it. So this is always part of what we ask to clinicians when we do the early evaluation of the value propositions at these early stages. And I will talk a bit more about that in the next slide, but we will have one talk on Friday on these. So these links with these care path analysis, the way in which we look at this is, so what is the care pathway is really the journey of a patient into the healthcare system, where we can understand what is happening now, what is the clinical need, and how your technology could help to solve an existing clinical need. Again, this is covered on Friday by Amanda from our MIG, and this feeds, and it's really important and inform the early economic modeling. In the early economic modeling is different from a full cost effectiveness analysis. Really, I think that it's an important step in the evaluation. The aim are different. It's not going to be the model that you're going to NICE to present to NICE. This is just to understand. Sometimes it could be a headroom analysis to understand the price point for the test. It could be the main aim of an early economic model could be understanding the inclusion criteria, for example, for your study, for your clinical studies, and it could be just to understand exactly what are the constraints for the use of your test in the clinical setting. So Joy and Owen will talk more about the difference between early economic modeling and cost-effectiveness analysis in the health economic webinar. So let's focus now on these clinical studies. So the first step, as I mentioned previously, is understanding what are the value proposition of your test. And this is really essential because it frames the whole journey of evidence gathering. Why is your test important? How do we do this? We do it with the qualitative data collection, usually interviews, more rarely surveys, if we've got specific questions, and we review the guidelines. So there's quite a lot of understanding to extract the pathway in the current pathway. And the outcomes are the role of the test. So where in the pathway the new test fits, and what's and what is the target population. I mean, these are the main outcomes, but Amanda will go much more in detail than this on Friday. So the next step, or usually these two steps happen in parallel. So while we do the value proposition study, usually the company look at analytical validity. So the main questions that these studies are asking is, does the test generate the expected results in ideal circumstances? Ideal circumstances mean the laboratory. So like in an ideal setting where everything is under control. You've got developers who are used to the test, using the test, so that it's unlikely you've got errors, links to misuse of the test. It's an ideal circumstance because usually you use retrospective samples collection where you select only the samples where definitely you've got the disease or definitely don't have the disease. Or you can use a case control studies where you are just selecting the black and white situations, not the gray in the middle that is characteristic of a clinical setting. If you don't have very good results in these ideal circumstances, it's very unlikely that progressing into the clinics, you would have better results, possibly impossible. So here is where you're really kind of redefining, working and reworking on your prototype, make sure it's actually getting the best possible chances to be successful in the next steps. Main outcome is accuracy, reproducibility, and safety. And there is much more on that on the paper, if you want to look at it. This is the first step and the first study in the clinic. So it's the diagnostic accuracy study where you want to look at clinical performance and you want to understand how well the test perform, but this time in a clinical environment. So the usual study design for these studies is a cohort observational study. So this means that cohort study means that you define some pre-specified inclusion criteria, and you are including everybody that who fits with those criteria and pass through your door. So you're saying I'm going to recruit patients with suspected stroke as determined by paramedics. Paramedic decide, this is a suspected stroke, go to the hospital, is included in that way. And then in the results will have to account for all of these patients who have been included. The primary outcome of these studies are sensitivity and specificity and predictive values, and we've got a webinar on that explain everything about this, so I'm not going to spend time on it, but Kevin and Claire will really talk you through how to calculate sensitivity and specificity and predictive values. Just the last consideration about this is these studies are observational. What does it mean? It means that usually you're blinded to the clinicians, and the patients are blinded to the results of the study. There are a couple of main reasons for that. The first reason is that you don't know if the test works, so you don't want to influence the clinician on the clinical decisions based on a test that you don't know if it works. So that's the really main reason. But it's important also for reducing bias and in the interpretation of the results. So the next step is the clinical utility study. Here, we've got different questions this time. So we now know that it works, in theory, no? That's what we looked at until now. It works also in a clinical environment. So we now want to understand if this test is actually useful in clinical practice. So it's a different kind of question, obviously, so you need a different kind of design. To understand if it's useful, you can't have an observational blind study. You need to actually use the test in the clinical environment that you've identified as the one that is more likely to benefit the patients. So you're doing an interventional study this time, so that means that the clinicians are unblinded to the results of the test. They're using the test in the way in which is defined in the protocol. So it's still like an ideal evaluation. It's not a real-world evaluation at this stage. You've got a strict protocol that clearly defines who you're recruiting, who is the person who is the patient population that can benefit from the study and how the test should be used very carefully. In the main outcomes here are the patient's outcomes, or if and whatever, I mean, the outcomes that have been defined here. So if you've defined a specific value proposition, if you think that reducing the time to diagnosis, for example, means that you're going to decrease the mortality of that population of patients, here is the time to actually record it. So you will have two groups. So one group will use the standard practice, and it's really kind of, you're comparing really two pathways, not just two tests. So one will be comparing the current pathway, one group, and the other one will be comparing the potential new pathway with the new test introduced into the pathway. And this is the time where you are looking at the difference between these two pathways and see if actually your outcomes in the new pathway are better, and the outcomes are defined as they are representing the value proposition that you identified. And this is really important. We have the conversation in the past with NICE, and they were saying, you know, often we've got this report of, you know, the summaries of evidence from companies, and they say this test is brilliant because it can do this, this, and this. And actually, when you go and look at the evidence they've got collected and the studies they've done, they haven't really found the evidence for what they claim. So that means that that claim is not being considered true or you know, reliable. So always be careful with matching the value proposition with the outcome and the data you're recording during the studies, because if you're not doing that, then that won't be accepted as evidence. Here is also the phase where you look at cost effectiveness. So you usually have got the full health economics at this stage, that it's linked to the outcomes. So that's really the moment in which you can compare the cost utility and of your test, and that could be the real you know, advantage of your test, that you can be decreasing cost, maintaining the same patient outcomes, for example. That could be a value in itself. Now we have really decreased the uncertainty linked to the test, and we are ready for adoption. Usually you've got older studies at this point that are usually smaller, but the main question is understanding if it's useful in that local context. So when you are introducing a test in real practice and you don't have a very strong protocol, like in an RCT, in a randomized controlled trial, for example, tests are not used always in the way in which they should be used. Perhaps they're used in a slightly wider population, and where it hasn't been completely demonstrated its validity and utility. So it's important to actually assess again, plus obviously pathways are all slightly different. So the comparison that you've used in clinical utility study might not be completely the same as that you've got in that specific local context. So usually CCGs before adoption wants to actually see how it works in their own CCG. But usually it's a small study, it's a real world evaluation. So with a more relaxed protocol, usually you can have a 5-page protocol compared to a 50-page protocol. So it is much more left to the interpretation of the clinician as it would be in real life. The main outcomes that you want to look at is the feasibility in that setting, and also the patient's staff experience is very important in this kind of evaluations. So last question for you, now that we know what we are talking about, and we can look at the understanding what really are the problems for you, and that's useful for us to understand how we can help you, and for designing the next webinar series. We've got some ideas of what are the hurdles, but obviously it's much better to hear from you to understand what are your the concepts that are more difficult to understand from a theoretical point of view, but also practically from an implementation point of view. You can see you're voting. And voted. %, almost there. Okay. I think we can stop. So the clinical utility study seems the one that are more difficult, but together with adoptions and implementation studies probably, and health economics also. Seems to be quite, the care path analysis also, seems to be, there are quite, and the evaluation evidence gaps actually, it was hidden there at the bottom. It seems to be the one that gives more problems. Yeah, I agree. I think that's the difficult one, to understand exactly where you are when life is not linear, as in my nice drawing, and actually everything is a bit more complex. So hopefully to understand exactly what are the different steps can help with that, and then obviously we're always happy to help and hear your experience if we can help you with some of these. So thank you for answering our last question. I'm now just going to show you the next webinars so that you've got a chance also to subscribe to some more of the webinars of the series. As I mentioned, if you can't join for a specific one, you can always find it on YouTube or on our webpage. So don't worry. So the link here on the top is where you can register for the other webinars. As I mentioned, on Monday is going to talk about the care pathway analysis. And this is really important. I really would advise you to join this webinar. Understanding what are the value proposition of your test, as I mentioned already, is really crucial and help you to understand also what are the next steps and what is the right strategy for the evaluation. You know, when you're saying that it's hard to understand the whole pathway and what is the overall evidence you need, usually it's hard because you haven't identified very, very strongly the value propositions at the beginning, because everything else should be around that. Everything else should be finding the evidence for those value propositions. So obviously, if those are not clear in the mind of the developer and the evaluator, then it's really hard to actually find the right strategy to get those evidence. And this is going to be on Friday. Then the next one next week is on diagnostic test evaluation with Kevin, who is from Maths, Stats and Physics here in Newcastle University, and Claire within our MIC. Kevin is really a good speaker, and if you are not sure about sensitivity and specificity and predicted values, just join them, because they're really, really clear explanations there about the statistics. And they also cover an imperfect reference standard that is very common problem in diagnostics. So next week is all about statistics, and we've got James who works in the university, and his talk is going to be mainly on study design, specifically focusing on clinical utility study, but we'll also try to squeeze something on sample size calculations for diagnostics. So everything you want to know about study design, you can go to his webinar. We've got a session on systematic literature reviews. It's really important to understand this concept. Sometimes you don't need a full systematic literature review. And they are going, Ryan and patients are from the university, the Institute of Health and Society, and probably previous Institute of Health and Society. They've changed all the names just to confuse me. But they can explain you why you want to do a literature review in a systematic way. Why it's not a very good idea to cherry pick your evidence so that you can make your own story, but actually that's not reflecting potentially what it's out there in the literature. So it doesn't have to be a 1 year, project you can do a rapid systematic review, and I show the difference between these two. That still is systematic and is much quicker. So go, and they will have some tips for you to help to understand how to be systematic and rapid at the same time. Lisa, she works with us very closely. She's great. She's from the Stroke Research Group, and her talk is usually very appreciated by everybody, because very, very clearly she will explain you the whole practical aspect of actually doing a clinical studies. So it's from a summary from funded application up to study closure, everything, all the difficulties, the hurdles, the hoops you have to jump through, time scales, so that you can plan your studies appropriately, and that's really important when you're writing a grant application. Make sure that you're not underestimating how long these things take. Make sure you know who to talk with when. If you've got any of these questions and you're not sure about this, just go to Lisa's webinar, and that's what this is all about. Then we've got Kasia, who joined us last year, and she's been really great and helpful understanding how we can engage the patients and the public in the work we do. Now, I have to say, with the COVID situation, the diagnostics have become much more a discussed topic. Everybody now knows about PCRs and serological tests, and so everybody's an expert, but it wasn't like that before, and the involvement of patients and public was really important. So she will give you a lot of tips, practical tips, if you want to engage with patients and public, when, how. So all our webinars are very practical, and hopefully you can get some helpful tips for your research. Then we've got health economics evaluation, usually a very popular topic. Joy is the other senior methodologist with me in the MIC, and Owen is again from the evidence synthesis group. Has been working with us very closely on a few different studies, and is very interested in diagnostic evaluations for health economics. They will be describing the difference between the economic modeling and the cost-effective analysis. And finally, we've got a guest speaker, Basilakra, is the first time he's presenting with us in this webinar. We invited him because last year we had clear feedback that there was a need for clarification about the regulation for MDR and IVDR. So we invited Basil because we went to one of his talks, and he's really an engaging speaker, and to be engaging about regulations, I mean, it's quite hard, but he was great, and he really made the topic fun. And so we decided to invite him. He's been, he just opened his consultancy company, but he's been in working in the biggest notified body in Europe for more than 10 years. So any questions on regulation, just ask him. That's going to be the last one, the last webinar of our series. So please give us feedback, because we actually implement the feedback you give us, and if there is a clear indication for a need for a specific topic to be covered, we're very happy to include this in the next series. So thank you very much. Here there are few information about the LinkedIn and the email addresses if you've got questions in the next days, months. So now I have to understand how to take the questions. I'm quite new to all these so you can see the questions here on the chat. Rachel, do you want to unmute yourself and possibly reading some of the questions?
Yeah, yeah. So there's been a few people asking about links to the webinar series and links to the paper that you mentioned. So I've just provided those. And then we just received a question around whether we'll be covering the NICE diagnostic assessment program in our sessions. I think some of this will be covered in the health economic session. So Joyce just suggested that, that they might want to come to that session.
Yeah, it would be, it's a good idea perhaps to have one specific session on that for the next time, potentially, you know, where we describe all the NICE process in depth. We used to do something similar in the past. Yeah, we don't have a specific session just on the NICE processes. But yeah, we cover the requirements for different programs, the requirements for health economics for different programs in Joyce and Owen's session. Any other doubts?
Yeah, I don't think there's anything else right this second, but maybe while people are maybe thinking, yeah, what do you think are the major challenges for particularly small companies that are developing medical devices?
So, I mean, we had the poll and on that topic. I think that yeah, as I mentioned, I think matching the value proposition so with the design of the study it's really hard, because it's one of the things I just kind of understood almost like recently, like how much you know, the population, the value proposition, you have to be very, very careful to how they are worded. It's not just you know, we can provide the results more quickly. It's actually, you need to kind of define the population, so which patients specifically, you know, not just COPD patients, but I don't know, COPD patients who had more between 2 and 5 exacerbations per year, for example. I mean, it depends on the technology, but sometimes you have to be specific. And if you manage to be so specific, for example, using early economic modeling, then you've got almost your protocol for the clinical studies written. And that really helps, because then, you know, your patient inclusion criteria are going to be the population of interest. Your outcome in the clinical utility are the actual value propositions. And then it's much easier not to miss something, to record during the studies. So I think that's an important thing that I've seen sometimes not considered and included enough. But, so I think there are a few questions coming through.
Yes, so there was a question around, again, in the health economics session, will value of information analysis be covered?
I think so. Joy, you are here in the audience, right?
Yeah, It is covered very briefly. I think we actually took it out during the last webinar because we thought it might be too much detail, but I can make sure that it's covered briefly. Obviously, it's very difficult to go into details of all the different types of analyses that you use in health economic studies, but we'll make sure we specify how and when each are useful, and we'll make sure we add in a bit about value of information analysis into that.
Yeah, we can also provide some links for further information if you want to go deeper into a topic, because it's hard to cover in an hour everything. So,
and actually, linked to your point, Sarah, our paper is probably a good place to go to for that as well, for links to various different places where analyses are covered in more detail from all of the different points in evidence development.
Yeah, the paper has got a lot of links also to Shiny apps. So again, there are quite a lot of practical tips that can be helpful for developers, and links to calculators for sample size and other stuff. There is quite a lot. So if you go through it, there might be more useful information in there too. So the next question. To test a novel device, does the IP need to be shared with the NIHR the trust? I mean, I guess you mean not to test, to kind of to develop. So if you're developing a new device, and if you work in the trust, I mean, if you're not working in the trust, then you've got your own company. The IP stays with the company. I'm not an IP expert, to be honest, but that's what I've learned working in the MIC. If you work in the trust and you are developing a new device, yes, you should talk with the business development manager in the trust to understand how to protect your IP and make sure that you're not publishing papers before protecting the IP of your new invention. But yeah, that's probably a discussion you should have with the BDM in the trust or in the university, depending where you're based. If you are a company, I'm sure that you're quite switched on with the IPs, but the IP stays with the company. And when we work with the companies, we've got, we keep the IP of the research, but we don't, but the IP of the technology stays with the company, obviously. Do you always help developers to generate evidence for UK-based diagnostics, or are any developers aiming for other settings? So recently, actually, we've got a couple of projects that where there is an interest to look at the US, especially. So we are doing evaluations of capacity analysis for US. So yes, we are. Our remit is UK because we are an NIHR infrastructure, so that's our main aim, and we are focused on that. But yeah, recently we are working with the US in collaboration with some companies that do this kind of work in the US. Because obviously it's all a new, different system, so we feel it's better to start working with somebody who knows the system well. Why the group is focused on in vitro? I think everything in the webinar series at least applies to in vivo diagnostic too. I work in imaging and I haven't come across the MIC before, but I think it could be useful. Yes, definitely. I should have said that we work in vitro diagnostics so that's the remit for the NIHR, but outside the remit for the NIHR that we've got, we also work on imaging. I've got a project on breast cancer screening through imaging, for example, and medical devices in general, broadly. There are a lot of overlaps, yes, but that's the remit for which we were creating the NIHR. So, you know, but we are aware that a lot of this fits, and it's useful to a wider range of tests. So yeah, thank you. Any more questions? I'm sure you will have a lot more questions for the specific session. So this is really just an overview to make sure that you know how everything fits in and why each session has been chosen to cover that specific topic. I think we just had a couple of themes that we're not covering with this specific webinar. I think one is adoption studies and another one is the analytical validity. We're thinking to potentially add webinars to cover those two topics in the future. So if there are no other questions, we are just 3 minutes before 11. So I guess we can thank you and please fill the feedback forms so we can hopefully next time design it even better and more aligned with your needs. I hope that was helpful and clarify a bit what we are doing and why. See you in the next sessions. I'll probably be joining most of them, so I'm sure we'll talk again in the future. Thank you very much for your feedback.